You think you know what life looks like? Cells under a microscope, bacteria in a petri dish, maybe a virus under the electron microscope beam. But step past the textbook diagrams and you hit microbes that rewrite every rule. Magnetotactic bacteria with internal compass needles made of pure magnetite. Archaea, thriving in acid, hot enough to dissolve steel. Giant viruses larger than some bacteria with genomes holding over 2,000 genes. Prions that are not even alive but kill anyway. No DNA, no RNA, just twisted proteins spreading like a plague through brain tissue. Which ones are real and which ones sound like science fiction? All of them sit in lab cultures right now. Documented in peer-reviewed journals, sequenced down to the base pair. These are not legends. They're inhabitants of Earth today, and they have been here longer than we have. Some survive in environments that would kill you in seconds. Others live inside you right now, invisible passengers you will never meet. The line between living and non-living blurs when you look close enough at these things. Let us walk through the strange gallery of microbial life, piece by documented piece, and see what actually exists when you turn over the rocks. Science forgot to check for decades. Bacteria, archaea, protists, fungi, viruses. Everyone learns these categories in school. Simple boxes. Clean divisions. Then the discoveries started piling up. 1992. A French team investigating a pneumonia outbreak in England pulls water samples from a cooling tower. Inside the water, they spot something odd infecting acanthamoeba, polyphaga amoebas. It looks like a bacterium under the light microscope. Large, gram-positive staining, but it is not a bacterium. 11 years pass before anyone realizes what they found. 2003, La Scola and the team at the University of the Mediterranean published the truth. It is a virus, a giant virus. They name it mimivirus, short for mimicking microbe, because it fooled everyone into thinking it was bacterial. The capsid measures 500 nanometers in diameter. Add the protein filaments projecting from the surface and you hit 600 to 700 nanometers total. That is bigger than some bacteria. The genome, 1.2 million base pairs, 979 protein-coding genes, amino acid transfer RNA synthetases, enzymes science thought only cellular organisms could produce. The virus replicates entirely in the host cytoplasm, never entering the nucleus AO, which breaks another rule for DNA viruses. 2011, megavirus chilensis turns up in seawater off the Chilean coast. Capsid diameter 440 nanometers. Genome 1.26 million base pairs. 7 amino acid transfer RNA synthetases instead of 4. Science keeps finding more. 2013, the Pandoravirus family appears. Pandoravirus salinus from sediment near a Chilean river mouth. Pandoravirus dulcis from mud in a freshwater pond close to Melbourne, Australia. These are not just big, they are enormous, 1 micrometer long, 0.5 micrometers wide. Oval shaped with 3 membrane layers, the genome runs 2.5 million base pairs for Pandoravirus salinus. That is larger than some parasitic bacteria. About 93% of Pandoravirus genes match nothing in any database. No bacteria, no archaea, no eukaryotes. Some researchers speculate these viruses represent a 4th domain of life, though others call that premature. Either way, the genes are there, sequenced and verified. Published in Science in July 2013 by Jean-Michel Claverie and Chantal Abergel from Aix-Marseille University. Patrick Scheid in Germany found one in 2008 out. Living in an amoeba inside a woman's contact lens during a keratitis case, but he thought it was an endocytobiont, not a virus DAO because of the sheer size. If you value documented microbial discoveries and viral evolution history like this, subscribe. It is the best way to support our work. Also, comment from which city you are hearing this from. Also, this script is researched and factual, using sources from Biology LibreTexts, published January 28. 2025, including OpenStack's microbiology textbooks, and the peer-reviewed journal NPJ Viruses, published February 11. 2025, detailing 20 years of giant virus research with electron microscopy data and genome sequences from institutions, including the French National Research Institute for Sustainable Development. We are not guessing. We are reading the lab reports. Now back to the information. Giant viruses shatter the definition of what a virus should be. Traditionally, viruses are small, simple, entirely dependent on host machinery. Mimivirus and its relatives carry their own transcription apparatus. They package enzymes inside the viral particle before infection even begins. The replication cycle spans 10 to 15 hours. The virus enters through phagocytosis. The internal core, a dark nucleoid visible under electron microscopy, stays detectable in the cytoplasm after extrusion from the particle. Viral factories grow from 0.4 micrometers to 10 micrometers in diameter, large enough to be infected by their own parasites. Yes, viruses that infect viruses. Viriphages. La Scola discovered the first one in 2008, Sputnik virophage. It replicates inside the mimivirus factory and reduces mimivirus yields. Of the genome, 18,000 base pairs, 21 genes. It cannot replicate without the giant virus, a parasite of a parasite. Turn from viruses to bacteria, and you hit organisms with internal navigation systems. Magnetotactic bacteria, discovered twice. First, in 1963 by Salvatore Bellini at the University of Pavia in Italy. He examines bog sediments under his microscope and sees bacteria swimming in one direction, consistently northward. He calls them magnetosensitive bacteria. He publishes in Italian in the official journal of the Istituto di Microbiologia. The publications sit unnoticed. 1975, Richard Blakemore rediscovers them independently in freshwater near Woods Hole, Massachusetts. He shows the bacteria orient along magnetic field lines. They move via magnetotaxis. Though the term is misleading, this is not a stimulus response like most taxis. The bacteria contain fixed magnets. Even dead cells align like compass needles. The magnets are magnetosomes. Membrane-bound organelles containing crystals of magnetic minerals, magnetite, chemical formula iron 3, oxygen 4, or graphite, iron 3, sulfur 4. The crystals form chains inside the cell. Each crystal measures 35 to 120 nanometers, within the single magnetic domain size range. The magnetic dipole moment of the cell equals the sum of all magnetosome dipoles. Enough to passively orient the bacterium and override thermal motion. The bacteria thrive at the oxic-anoxic interface, the transition zone between oxygen-rich and oxygen-starved water or sediment. Many species survive only in very limited oxygen. Some exist only in completely anaerobic conditions. The evolutionary advantage? Simplifying a three-dimensional search for favorable chemical conditions into a one-dimensional movement along the magnetic field. Instead of wandering randomly through water, the bacteria follow field lines down toward the sediment where they find the oxygen levels they need. Magnetotactic bacteria span multiple phyla: alpha proteobacteria, gamma proteobacteria, delta proteobacteria, nitrospirae. Most cultured species are microaerophiles or anaerobes. They grow chemolitho-autotrophically using reduced sulfur compounds or chemo-organoheterotrophically using organic acids. Recent work in 2024 shows some are extremophiles. Lefebvre and colleagues described magnetotactic bacteria in hot springs in northern Nevada with upper temperature limits around 63 degrees Celsius. Others isolated obligately alkalophilic strains from Mono Lake, California. A hypersaline and extremely alkaline environment. Optimal growth pH of about 9.0. The bacteria also appear in deep-sea hydrothermal vents, where they face high pressure and temperature simultaneously. The strangest magnetotactic bacteria are the multicellular ones, Magnetoglobus species. Multicellular magnetotactic bacteria, abbreviated MMB. They are the only known bacteria without a unicellular stage. Groups of several tens of cells physically attach together and behave as a single organism. Consortia, spherical, 3 to 8 micrometers in diameter, a monolayer of cells. Each cell has tens of magnetosomes and about 30 flagella covering the outer surface. New consortia form when existing ones divide symmetrically. Individual bacteria never join or leave existing consortia. July 11. 2024, Schabel and colleagues published in PLOS Biology that MMB consortia are not clonal. They separately sequenced the metagenomes of 22 individual consortia, representing 8 new species. The cells within each consortium are genetically heterogeneous. The paper shows metabolic differentiation. Some cells specialize in different functions within the same consortium. The work comes from Montana State University, the Department of Energy Joint Genome Institute, and Marine Biological Laboratory using single-cell genomics and nanoscale secondary ion mass spectrometry. These consortia live in Little Sippawisset Salt Marsh near Falmouth, Massachusetts, and between sand grains. The sand grain diameter averages 0.6 millimeters. Random packing creates pores with typical radius 0.2 millimeters. The local geomagnetic field measures 50.9 microteslas with an inclination of 65.6 degrees. Magnetotactic bacteria produce magnetofossils that preserve in the geological record. The oldest unambiguous magnetofossils come from Cretaceous chalk beds in southern England. Less certain reports extend to 1.9 billion years old in the Gunflint Chert. Some researchers claim magnetite particles in the Martian meteorite ALH 84001 match magnetosome morphology, suggesting ancient Martian life, but this remains highly contested. Step into extreme environments and you find archaea that laugh at conditions lethal to most life. Thermophiles, heat lovers. Thermus aquaticus thrives between 41 and 70 degrees Celsius. Discovered in hot springs at Yellowstone National Park. The heat-stable DNA polymerase from Thermus aquaticus, called Taq polymerase, revolutionized molecular biology. It made polymerase chain reaction, PCR, practical. Without Taq polymerase, modern genetics collapses. Hyperthermophiles go further. Pyrodictium occultum and Pyrococcus abyssi survive above 80 degrees Celsius. Some archaea hit 110 degrees Celsius in deep-sea hydrothermal vents. The proteins resist denaturation through specific amino acid composition, increased salt bridges, and compact folding. Thermoplasma acidophyllum combines heat tolerance with acid tolerance. It lives in coal refuse piles where temperatures hit 60 degrees Celsius and pH drops below 2. Psychrophils, cold lovers, they thrive at 15 degrees Celsius or lower. Chryssobacterium greenlandensis grows in glaciers. Methanogenium frigidum lives in Ace Lake in Antarctica. Temperatures this low normally kill bacteria by slowing enzyme activity and forming ice crystals that rupture cells. Psychrophiles adapt with antifreeze proteins that protect DNA. The proteins prevent ice formation. Membrane lipids stay fluid at low temperatures through increased unsaturated fatty acid content. A 2022 paper by Kashyap defines psychrophiles as organisms with optimal growth at 10 degrees Celsius and some growth at 0 degrees or below. Psychrotolerant microorganisms differ. They grow at 0 degrees but prefer 20 to 40 degrees. New terminology emerged. Uropsychrophiles, cold lovers that also grow well in the mesophile range. Stenocycrophiles, strictly cold lovers. Psychrophile enzymes work in detergents and bioremediation in cold environments, cleaning toxins from soil and water at temperatures where other enzymes fail. Halophiles, salt lovers, extreme environments with high salt concentrations. Halobacterium, halobium survives in the Dead Sea and Great Salt Lake. Hortea wernicki tolerates salt. Tetragenococcus halophilus grows in soy sauce and salted anchovies, adapting to 35% salinity. High salt normally kills bacteria through osmotic effects. Water drains from the cell, causing dehydration and death. Halophiles adapt by pumping salt from their cytoplasm or accumulating compatible solutes, like glycine betaine. Their proteins resist denaturation in hypersaline conditions through extensive negative surface charges. The charges create hydration shells that prevent aggregation. Haloalkaliphals, like Neutronomonas ferronis, combine salt tolerance with alkalinity tolerance, growing at pH above 11. They live in soda lakes, where sodium carbonate dominates. Recent work in 2024 from CAS identifies halophiles like Halomonas elongata, sequestering heavy metals like cadmium from saline industrial waste. Bioremediation in environments too harsh for conventional microbes. Acidophiles, acid lovers. They require pH less than 5. Acid Ethiobacillus ferrooxidans extracts heavy metals like copper and uranium from mining waste through bioleaching. It oxidizes ferrous iron and elemental sulfur, generating sulfuric acid. The organism thrives at pH below 3, found in acid mine drainage where sulfide mineral oxidation creates extreme acidity. Some acidophiles grow at pH 0.7. Their cell membranes contain unique lipids that prevent proton leakage. They pump protons out continuously to maintain a cytoplasmic pH around 5.0 to 6.5. Picrophilus torridus, a thermoacidophile, grows optimally at 60 degrees Celsius and pH 0.7. Found in volcanic sulfiteric fields in Japan, the proteins show increased negative surface charge and thermophilic adaptations. Acidophile proteins work in metal extraction, making bioleaching economically viable. Alkaliphals thrive at pH 9 to 11. Bacillus alkalophilus grows in alkaline soils and soda lakes. The challenge? Maintaining proton motive force when external pH exceeds internal pH. Alkaliphals adapt through sodium proton antiporters and unique ATP synthase variants. Their enzymes, alkaline proteases and lipases, work in detergents and industrial processes, textile processing, paper production. The enzymes remain stable and active at pH levels that destroy standard enzymes. Barophiles, or piezophiles, pressure lovers, deep-sea microbes experiencing hundreds of atmospheres, 1 atmosphere equals 1.3 kilopascals. The Mariana Trench bottom hits 1,000 atmospheres. Microbes there adapt with pressure-resistant membranes containing polyunsaturated fatty acids. Proteins fold differently, maintaining function under compression. Many deep-sea piezophiles are also psychrophiles and thermophiles if near hydrothermal vents, combining multiple extremes. A 2017 study by Pauly and Frontiers in Microbiology examines piezophiles from deep-sea sediments, showing they modify lipid composition and protein structures to resist pressure denaturation. Radiophiles resist radiation. Deinococcus radiodurans survives radiation doses 5,000 times higher than what kills humans. 15,000 grays. The bacterium repairs double-strand DNA, breaks through redundant genome copies and efficient repair enzymes. Found in medical equipment sterilized by gamma radiation, where it survives and grows. The pink pigmented colonies appear in supposedly sterile environments, shocking microbiologists. Discovered in 1956 by Arthur Anderson at Oregon Agricultural Experiment Station, studying canned meat that spoiled despite radiation treatment, beyond extremophiles lie microbes that defy categorization. Candidate phyla radiation, abbreviated CPR, nano-sized bacteria forming over 15% of bacterial diversity. They are mainly symbiotic, often predatory. Cells measure around 200 to 300 nanometers, smaller than some viruses. They lack many essential biosynthetic pathways, depending on hosts for survival. Recent work in 2024 from the French National Research Agency shows CPR bacteria associated with magnetotactic bacteria, partially inside the host cell. The magnetotactic bacteria modify their external membrane in response to CPR contact. The CPR genomes reveal streamlined metabolisms, minimal gene sets, and unusual ribosomal RNA structures. Most remain uncultured. Discovered through metagenomics when researchers sequence environmental DNA directly without culturing organisms first. Norman Pace pioneered this in 1980, showing microorganisms could be identified in natural populations without culture by amplifying ribosomal RNA genes using PCR with specific primers. Turn away from bacteria and archaea toward the acellular agents, viroids, plant pathogens. Small, naked, single-stranded circular RNA molecules, no protein coat, no capsid, just RNA, discovered in 1971 by Theodore Diener at the Agriculture Research Service while investigating potato spindle tuber disease. He names them viroids, meaning virus-like. The potato spindle tuber viroid, PSTVD, measures about 360 nucleotides. It spreads when infected knives cut healthy potatoes during planting. Symptoms include spindle-shaped tubers, stunted growth, reduced yield. Other viroids followed. Tomato planta, macho viroid, TPMVD, causes chlorophyll loss, brittle leaves, tiny tomatoes. Avocado sunblotch viroid, ASBV'd, lowers fruit yields and quality. It is the smallest viroid discovered in plants. Peach latent mosaic viroid, PLMVD, causes necrosis of flower buds and branches, wounding ripened fruit, allowing fungal and bacterial growth. It affects plums, nectarines, apricots, cherries. Agricultural losses reach millions of dollars annually. Viroids infect through wounds or contaminated tools. They replicate using host RNA polymerase. The circular RNA folds into complex secondary structures stabilized by base pairing. Some viroids have catalytic activity, acting as ribozymes. They do not code for proteins. Their entire pathogenic mechanism relies on RNA structure interfering with host gene regulation. Human viroid diseases remain undiscovered. One exception, hepatitis D, the hepatitis delta virus or viroid, depending on classification. It requires hepatitis B virus to provide the capsid for entry into liver cells. The delta agent has circular single-stranded RNA, about 1,670 nucleotides. It codes for one protein AIDS delta antigen. The viroid enters through the hepatitis B surface antigen coding. Once inside, it replicates autonomously in the nucleus using host RNA polymerase. Discovered in 1977 by Mario Rossetto in Italy, published in Gut, the viroid spreads via blood transfusions and can cause fulminant hepatitis. 3 tests diagnose it: immunofluorescence on tonsil biopsies, Western blot analysis of blood tissue, capillary electrophoresis of blood. If you value documented infectious agent research and molecular pathology like this, subscribe. It is the best way to support our work. Also, comment from which city you're hearing this from. Also, this script is researched and factual. using sources from Biology, Libre Texts, updated April 21, 2024, on viroids and prions, and the peer-reviewed journal Viruses, published February 26, 2024, by Steger, Reisner, and Stanley Prusner on subviral pathogens, documenting decades of research from Heinrich Heine University, Dusseldorf, and University of California, San Francisco. Now back to the information. Prions, proteinaceous infectious particles, the strangest agents of all, no DNA, no RNA, just protein, infectious protein. Stanley Prusner proposed the concept in 1982 while studying scrapie, a fatal degenerative disease in sheep. He shows the infectious agent resists treatments that destroy nucleic acids. Ultraviolet light barely affects it, unlike viruses. Chemical treatments that destroy proteins do destroy infectivity. He concludes the agent is protein-based. The scientific community responds with skepticism. Infectious agents without nucleic acids violate the central dogma. How can information transmit without DNA or RNA? Prusner persists. 1997. He receives the Nobel Prize in Physiology or Medicine. Prions are misfolded versions of a normal cellular protein. Prion protein, abbreviated PRP, the normal form PRP with superscript Ca for cellular, exists in neural cell membranes. Function? Sequestering copper ions. The protein has alpha helices in its secondary structure. The prion form, PRP with superscript Sc for scrapie, has beta sheets instead. This single structural change makes it infectious. When PRP contacts PRPC, it induces the normal protein to refold into the disease form. Autocatalytic. One misfolded protein creates more misfolded proteins. They aggregate into plaques. The plaques accumulate in brain tissue, causing neurodegeneration. The brain develops vacuoles, giving it a spongy texture. Transmissible spongiform encephalopathies, scrapie in sheep. Affected animals scrape against fences compulsively, hence the name. Bovine spongiform encephalopathy, BSEIs in cattle. Mad cow disease, first detected in the United Kingdom in 1986. It spread through the practice of feeding cattle nervous tissue from other cattle in their feed. Rendering processes used high temperatures, but not high enough to destroy prions. Prions resist heat. They survive autoclaving at 121 degrees Celsius for 15 minutes, the standard sterilization protocol. They resist formalin, a fixative that inactivates most pathogens. They resist ionizing radiation. The protein misfolds, but contains no nucleic acid to damage. The UK banned the practice of feeding ruminant-derived protein to ruminants in 1988. By then, over 180,000 cattle were infected. The epidemic peaked in 1992 with almost 37,000 cases, 4.4 million cattle slaughtered in attempts to control spread, variant Creutzfeldt-Jakob disease, VCJD, in humans, acquired from eating beef from BSE-infected cattle. The first cases appeared in 1996 in the UK. The disease presents differently from classic Creutzfeldt-Jakob disease. Younger age of onset, median 26 years. Psychiatric symptoms first, anxiety, depression, psychosis, then neurological decline, ataxia, dementia, myoclonus, death within 13 months of symptom onset. 178 confirmed and probable cases in the UK through 2014. The prion crosses from cattle to humans by consuming contaminated tissue, or primarily brain and spinal cord. The infectious dose is unknown but thought to be small. Many countries banned importation of British beef. The economic damage to the UK beef industry exceeded billions of pounds. Kuru in humans in Papua New Guinea. Spread through ritualistic mortuary cannibalism in the forepeople. Family members consumed the deceased, including the brain, as a sign of respect. Women and children ate the brain more often than men. Kuru means trembling or shivering. Symptoms begin with headaches and joint pain. Coordination fails. Tremors develop. Walking becomes impossible. Emotional ability, pathological laughter, death within 1 year. Carlton Gajdusek investigated Kuru in the 1950s and 60s. He showed it was transmissible by injecting brain tissue from Kuru victims into chimpanzees. The chimpanzees developed spongiform encephalopathy. He received the Nobel Prize in Physiology or Medicine in 1976. The practice of mortuary cannibalism ended in the 1960s. Kuru cases declined sharply. The last victim likely infected before the practice stopped died in 2009. The incubation period can exceed 50 years. Classic Creutzfeldt-Jakob disease, CJD, occurs sporadically worldwide. Incidence about 1 per million population per year. Familial forms exist, caused by mutations in the prion protein gene on chromosome 20. Fatal familial insomnia, another prion disease, causes progressive insomnia, autonomic dysfunction, dementia, death within 7 to 36 months. Gerstmann-Streusler-Scheinker syndrome, a familial prion disease, causes ataxia, dementia, death within 5 years, all currently untreatable. No drugs stop prion replication. No vaccines exist. Diagnosis relies on clinical symptoms, MRI showing characteristic patterns, electroencephalography showing periodic sharp wave complexes, and protein 1433 in cerebrospinal fluid. Definitive diagnosis requires brain biopsy or autopsy showing spongiform changes and prion protein accumulation detected by immunohistochemistry. Growing evidence suggests other neurodegenerative diseases involve prion-like mechanisms. Alzheimer's disease. Amyloid beta and tau proteins misfold and aggregate, spreading from cell to cell. Parkinson's disease. Alpha-synuclein aggregates into Lewy bodies. Huntington disease. Mutant Huntington. Protein forms aggregates. Amyotrophic lateral sclerosis. Superoxide dismutase 1 and TDP43 aggregate. These are not infectious between individuals like classic prions, but within the patient, misfolded proteins, template misfolding of normal proteins, propagating pathology through the brain. A 2015 paper in Acta Neuropathologica by Jucker and Walker reviews the evidence for prion-like mechanisms in neurodegenerative diseases, showing protein aggregates extracted from diseased brains, seed aggregation when injected into animal models. Beyond prions lie the giants among protists. Slime molds, not really molds, not fungi. Protists, myxomycetes. They exist as single-celled amoebas that feed on bacteria. When food runs out, they aggregate into a multicellular slug that migrates toward light and heat. The slug transforms into a fruiting body on a stalk. The fruiting body releases spores. The spores germinate into new amoebas. The transition from unicellular to multicellular and back occurs within one organism's life cycle. Dictyostelium discoidum, the model organism, shows this beautifully. When starved, up to 100,000 cells stream together, forming a slug 2 to 4 millimeters long. The slug migrates, then differentiates. About 20% of cells become stalk cells, lifting the rest into a spore mass. The stalk cells die. The spore cells survive, waiting for better conditions. The decision of which cells become stalk versus spore involves communication through cyclic AMP pulses. Cells that start secreting cyclic AMP early become pacemakers, organizing the aggregation. Research by John Tyler Bonner, starting in the 1940s, established the life cycle details. Modern work by Joan Strassman and David Queller shows genetic conflicts within the slug, with some cell lines cheating by contributing more spores and fewer stalk cells. Protists include organisms of astonishing complexity. Paramecium, a ciliate, swims using thousands of cilia beating in coordinated waves. The cell contains 2 nuclei, a macronucleus handling daily functions, a micronucleus for sexual reproduction. During conjugation, 2 paramecia exchange micronuclei, mixing genetic material without fusion. The macronucleus disintegrates and reforms from the micronucleus. Paramecium also hosts endosymbiotic bacteria like Holospora and Catabacter. Some Ketobacter strains confer killer traits. The paramecium releases toxins that kill non-infected paramecia. The bacteria gain from the host's dominance, discovered by Tracy Sonneborn in the 1940s. This revealed cytoplasmic inheritance and endosymbiosis roles beyond mitochondria. Euglena, a photosynthetic protist with an eye spot and flagellum. It swims toward light using the eye spot to detect light direction, optimizing photosynthesis. In the dark, it becomes heterotrophic, consuming organic material. The chloroplasts originated from a secondary endosymbiosis, engulfing a green alga. The cell has both plant and animal characteristics, blurring the boundary. John Hogg in 1860 proposed the Protoctista kingdom to accommodate such organisms. Ernst Haeckel named it Protista in 1866. These classifications preceded modern phylogenetics. Today, molecular data place Euglena in the Euglenozoa clade, distant from plants. Foraminifera, marine protists with shells made of calcium carbonate or agglutinated sediment. Pseudopodia extend through shell pores, catching food. Forum shells accumulate on the ocean floor, forming forum ooze. Over geological time, they become limestone. The White Cliffs of Dover consist largely of forum shells from the Cretaceous period. From species diversity tracks, environmental conditions, paleontologists use forum assemblages to reconstruct ancient climates and ocean chemistry. The shells preserve isotopic ratios of oxygen and carbon, recording ocean temperatures and ice volume. The Paleocene-Eocene thermal. maximum 56 million years ago, shows up in forum isotopes as a sharp temperature spike. Forum studies by Tjeerd van Andel and others established the method in the 1950s and 60s. Radiolaria, another marine protist group, build intricate silica skeletons. The skeletons display geometric beauty, inspiring Ernst Haeckel's detailed illustrations in Kunstformen der Natur, published 1904. The silica shells sink after the organism dies, forming radiolarian ooze on abyssal plains. Over time, they lithify into chert. Radiolarian fossils date back to the Cambrian period, 540 million years ago, making them excellent biostratigraphic markers. Their diversity tracks with ocean productivity and climate. The skeletons resist dissolution better than carbonate, preserving in deep sea sediments where calcium carbonate dissolves. Turn to fungi, and you find organisms that defy expectations. Carnivorous fungi. The family Orbiliaceae includes species with specialized hyphae that trap prey. Nematodes crawling through soil encounter adhesive networks. The fungus secretes sticky substances. The nematode sticks. The fungus penetrates the nematode cuticle, digesting the interior. Arthrobatris oligospora forms constricting rings. When a nematode enters, the ring cells swell rapidly, in 1/10 of a second, tightening around the worm. The nematode cannot escape. The fungus invades and consumes. Discovered in 1988 by Barendijk, research continues on mechanisms. Some fungi produce toxins that paralyze prey before trapping. Others form three-dimensional nets. The diversity of trapping mechanisms suggests independent evolution multiple times. The fungi supplement nitrogen-poor soils by capturing animal protein. Lichens, symbiotic partnerships between fungi and photosynthetic partners. Algae or cyanobacteria. The fungus provides structure and protection. The photosynthetic partner provides carbohydrates. The two organisms function as one. Lichens colonize bare rock, secreting acids that weather the surface, creating soil over centuries. They survive extreme conditions: Antarctica, deserts, Arctic tundra, space. Yes, space. Lichen samples survived outside the International Space Station for 1.5 years, exposed to vacuum, temperature swings, and unfiltered solar radiation. The European Space Agency experiment EXPOSE-EA, conducted 2008 to 2009, showed Rhizocarpon geographicum retained viability and resumed photosynthesis after return. Lichens produce unique secondary metabolites. Eustenic acid has antibiotic properties. Vulpinic acid is bright yellow, used historically as dye. Some lichens bioaccumulate heavy metals, making them bioindicators of air pollution. Monitoring lichen diversity tracks environmental quality. Industrial zones have low lichen diversity. Clean air supports diverse communities. Mycorrhizal fungi form symbioses with plant roots. Arbuscular mycorrhizae penetrate root cells, forming branched structures called arbuscules. The fungus delivers phosphorus and nitrogen from soil to the plant. The plant supplies sugars from photosynthesis to the fungus. Over 90% of land plants form mycorrhizal associations. Without them, agriculture collapses. Ectomycorrhizae coat root surfaces without penetrating cells, common in temperate forests with trees like oak, pine, beech. The fungal network connects multiple trees, forming a wood wide web. Trees can transfer nutrients through the network. Mother trees support saplings. Dying trees dump resources into the network. Research by Suzanne Simard at the University of British Columbia from the 1990s onward maps these networks using isotope tracers. Carbon isotopes injected into one tree appear in neighbors through fungal connections within days. Cordyceps fungi infect insects, manipulating behavior. Ophiocordyceps unilateralis infects carpenter ants in tropical forests. The fungus grows inside the ant, consuming non-essential tissues first. It leaves the brain for last. The ant behavior changes. It leaves the nest, an unusual behavior. It climbs vegetation to a specific height, around 25 centimeters downwards where temperature and humidity suit the fungus. It bites the underside of a leaf, locking its mandibles in a death grip. The ant dies. The fungus consumes the rest of the ant and erupts from the body, sending a stalk through the ant's head. The stalk produces spores that rain down on the forest floor, infecting new ants. The process takes 4 to 10 days from infection to death. David Hughes at Penn State University studies the mechanisms. A 2011 paper in BMC Ecology shows the fungus synchronizes ant death and spore release to colony foraging times, maximizing transmission. The fungus produces alkaloids that alter ant neural activity, compelling the summit behavior. The ant essentially becomes a zombie, controlled by the fungus. Fungi also produce compounds useful to humans. Penicillin, discovered by Alexander Fleming in 1928, comes from Penicillium notatum mold. Fleming notices bacterial colonies near the mold die. He isolates the substance responsible, naming it penicillin. Howard Florey and Ernst Boris Chain develop it into a drug in 1940. It saves millions during World War II, treating infections that previously killed. Other antibiotics follow. Cephalosporins from Cephalosporium acremonium, tetracyclines from Streptomyces bacteria. The pharmaceutical industry mines microbes for bioactive compounds. Cyclosporin. An immunosuppressant not trained comes from Tulipocladium inflatum fungus, discovered in Norwegian soil in 1969. It revolutionizes organ transplantation by suppressing rejection without destroying all immune function. Microbes produce enzymes used industrially. Amylases break starch into sugars. Proteases digest proteins. Lipases split fats. Cellulases degrade cellulose. These enzymes from extremophiles work in conditions that denature standard enzymes. Thermostable amylases from thermophiles operate in high-temperature brewing and baking. Alkaline proteases from alkaliphiles clean in high pH detergents. Cold-active lipases from psychrophiles work in cold water washing. A 2024 paper in Food, Science and Nutrition by Swaminathan reviews extremophile enzymes in food processing, including lactase for lactose-free products and enzymes that reduce acrylamide in fried foods. Acrylamide forms from asparagine and reducing sugars at high heat. Asparaginase from extremophiles breaks down asparagine before frying, reducing acrylamide up to 90%. Microbes bioremediate pollution. Alkanivirax borcumensis degrades oil spills in cold marine environments like the Arctic. It metabolizes hydrocarbons into non-toxic compounds. Geobacillus thermodenitrificans treats oil-contaminated industrial effluents at high temperatures. Ideonella sakaiensis, discovered in 2016 in Osaka, Japan, breaks down polyethylene terephthalate plastic, PT. The bacterium produces an enzyme, PTase, that hydrolyzes PET into monomers. A 2024 study identifies a psychrophilic Ideonella strain producing cold-active PEtases, breaking down plastic in cold environments where most enzymes fail, published in CAS Insights. This matters because most plastic waste ends up in cold oceans. Enzymes that work at 10 degrees Celsius or lower could clean marine plastic pollution at scale. Microbes, cycle elements, globally, nitrogen fixation, atmospheric nitrogen, N2, is inert. Most organisms cannot use it. Nitrogen-fixing bacteria convert it to ammonia using nitrogenase enzyme. Rhizobium forms nodules on legume roots. The nodules house bacteria in low oxygen environments. The bacteria fix nitrogen. The plant supplies energy. The fixed nitrogen fertilizes the plant and surrounding soil. Industrial nitrogen fixation via the Haber-Bosch process consumes 2% of global energy and releases carbon dioxide. Biological fixation achieves the same result using only sunlight-derived sugars. Cyanobacteria also fix nitrogen, particularly in oceans where fixed nitrogen limits productivity. Trichodesmium forms massive blooms in tropical oceans, fixing nitrogen and supporting marine food webs. Without nitrogen fixation, protein synthesis halts, and life as we know it cannot exist. Carbon cycle. Photosynthetic microbes fix carbon dioxide into organic matter. Cyanobacteria, algae, and some bacteria perform photosynthesis. They produce oxygen as a byproduct. 3 billion years ago, cyanobacteria caused the Great Oxygenation Event, raising atmospheric oxygen from trace levels to 21% today. The oxygen allowed aerobic respiration, which extracts more energy per glucose molecule than anaerobic pathways. This energy surplus enabled the evolution of complex multicellular life. The evidence sits in banded iron formations. Sedimentary rocks with alternating layers of iron oxide and silica formed when oxygen reacted with dissolved iron in oceans. The formations date to 2.5 to 1.8 billion years ago. Sulfur cycle. Sulfur oxidizing bacteria convert hydrogen sulfide and elemental sulfur into sulfate. Sulfate-reducing bacteria reverse the process in anaerobic conditions. Deep-sea hydrothermal vent communities rely on chemosynthetic bacteria oxidizing hydrogen sulfide. The bacteria form the base of the food web. Giant tube worms, Riftia pecaptila, host sulfur-oxidizing bacteria in their trophosome organ. The worms have no mouth or gut. They depend entirely on bacterial symbionts. The bacteria fix carbon using energy from sulfide oxidation. The worm supplies the sulfide, absorbed through its gills from vent fluid. Discovered in 1977 during Alvin submersible dives to the Galapagos Rift, the findings shocked biologists. An entire ecosystem independent of sunlight, powered by chemistry, methane cycle. Methanogens produce methane from carbon dioxide and hydrogen or from acetate. Methanogenic archaea live in anaerobic environments: swamp mud, ruminant intestines, termite guts, rice paddies. Methane, a potent greenhouse gas, contributes to climate warming. Ruminants produce about 120 kilograms of methane per cow per year via methanogenic archaea in their rumen. Global cattle population exceeds 1 billion. The agricultural contribution to atmospheric methane exceeds 30%. Methanotrophs consume methane, oxidizing it to carbon dioxide. Methylococcus capsulatus lives at methane seeps, limiting methane release. The balance between methanogenesis and methanotrophy regulates atmospheric methane levels. Phosphorus cycle, no gaseous phase like other cycles. Phosphorus moves through weathering of rocks. Bacteria solubilize phosphorus from minerals, making it bioavailable. Phosphate solubilizing bacteria secrete organic acids that dissolve phosphate minerals. Plants absorb the solubilized phosphate. Without these bacteria, phosphorus remains locked in rocks, unavailable. Phosphorus limits productivity in many ecosystems. Adding phosphorus fertilizers increases crop yields, but also causes eutrophication when runoff enters waterways. Algal blooms result, depleting oxygen and killing fish. The dead zones in the Gulf of Mexico and other coastal areas stem from excess phosphorus and nitrogen from agricultural runoff. Microbes also live inside us. The human microbiome. Each person carries trillions of microbial cells. Bacteria outnumber human cells. The gut microbiome alone contains 100 trillion cells representing thousands of species. They aid digestion, synthesize vitamins, train the immune system, and prevent pathogen colonization. Germ-free mice, raised without microbes, show abnormal immune development, thinner gut walls, and altered behavior. Fecal microbiota. Transplant treats a Clostridium difficile infection, a severe diarrhea resistant to antibiotics. Transplanting stool from a healthy donor restores normal microbiome, curing the infection in 90% of cases. First successful case reported in 1958 by Ben Eisman. But the procedure gained acceptance only in the 2010 decade after clinical trials. Microbiome composition associates with health and disease. Inflammatory bowel disease links to reduced microbial diversity. Obesity associates with altered firmicutes to Bacteroidetes ratio. A 2006 paper in Nature by Jeffrey Gordon's lab shows transferring gut microbiota from obese mice to germ-free mice causes the recipients to gain more fat than those receiving microbiota from lean mice on identical diets. The microbiome extracts more energy from food. Mental health also connects. The gut-brain axis allows microbes to influence neurotransmitter production. Lactobacillus and Bifidobacterium species produce gamma-aminobutyric acid, a neurotransmitter. Altered microbiome composition correlates with depression and anxiety. Whether causative or correlative remains under investigation, but animal models show behavioral changes from microbiome manipulation. Microbes also cause disease. Yersinia pestis, the bacterium causing plague, spread by fleas from rodents. The Black Death in the 14th century killed 30 to 60% of Europe's population. Genetic analysis of plague victims. Teeth confirms Yersinia pestis. Modern antibiotics treat plague if caught early, but untreated pneumonic plague kills within days. Mycobacterium tuberculosis causes tuberculosis. 1/4 of the global population carries latent tuberculosis. About 10 million new active cases occur annually. Drug-resistant strains spread. Multi-drug-resistant tuberculosis requires 18 to 24 months of treatment, with second-line drugs causing severe side effects. Extensively drug-resistant tuberculosis resists even more drugs, leaving few treatment options. Plasmodium species cause malaria, transmitted by Anopheles mosquitoes. The parasite infects liver cells, then red blood cells. Symptoms include fever, chills, anemia. Severe cases cause cerebral malaria, organ failure, death. 200 million cases annually. Over 400,000 deaths, mostly children under 5 in sub-Saharan Africa. Artemisinin-based combination therapies treat malaria effectively, but resistance emerges in Southeast Asia. The parasite's complex life cycle between mosquito and human complicates vaccine development. RTS, S vaccine shows partial efficacy, about 30% reduction in severe malaria, approved for use in 2021. Entamoeba histolytica causes amoebiasis, 50 million infections per year, 70,000 deaths. The amoeba invades the intestinal lining, causing dysentery. It can spread to the liver, forming abscesses. Metronidazole treats the infection. Prevention requires clean water and sanitation. Trypanosoma. Brucei causes African sleeping sickness. Transmitted by tsetse flies, the parasite crosses the blood-brain barrier, causing neurological symptoms. Confusion, sleep disturbances, seizures. Untreated, it is fatal. Treatment in late-stage disease involves arsenic-based drugs with high toxicity. Newer drugs like fexinetazole, approved 2019, offer safer alternatives. The study of microbes continues revealing surprises. Every environment sampled contains uncultured diversity. Metagenomics shows most microbial species have never been isolated. Deep sea sediments, polar ice cores, desert soil. Each harbors unique communities. Some estimates suggest millions of bacterial species exist. Only thousands have been cultured and characterized. The uncultured majority represents unknown metabolic capabilities and biotechnology potential. Synthetic biology engineers microbes for human purposes. Bacteria producing insulin for diabetes treatment. Yeast fermenting artemisinin precursors for malaria drugs. Engineered Escherichia coli synthesizing spider silk proteins. The possibilities expand as gene editing tools improve. CRISPR-Cas systems, originally discovered as bacterial immune systems against viruses, now allow precise genome editing. Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize in Chemistry in 2020 for CRISPR development. The system uses guide RNA to direct Cas nucleus to specific DNA sequences, cutting and allowing edits. Microbes engineered with CRISPR produce biofuels, pharmaceuticals, and industrial chemicals, replacing petroleum-based processes. Microbes may hold the key to extraterrestrial life. Mars once had liquid water. The Curiosity rover finds ancient lake beds in Gale Crater. Jezero Crater contains river delta deposits. If life arose on Mars, it likely resembled Earth's microbes. Extremophiles demonstrate life's resilience. Archaea thrive in conditions matching early Earth and potentially Mars. Magnetotactic bacteria could survive Martian subsurface brines. Radiolarians resist radiation. Mars lacks a magnetic field or thick atmosphere to block. Detecting biosignatures on Mars or icy moons like Europa and Enceladus relies on microbial metabolism signatures. Methane plumes on Mars could indicate methanogenic archaea. Phospholipids in Enceladus plumes could signal cellular life. The search for life beyond Earth is really a search for microbes. A 2023 paper in ISME Communications examines extremophile survival in simulated Martian conditions. Deinococcus radiodurans survived simulated Mars radiation and desiccation for extended periods. Halobacterium salinarum tolerated Martian salt concentrations and low temperatures. Bacillus subtilis spores resisted vacuum and ultraviolet radiation. The results suggest microbial life could survive in Martian subsurface refuges where conditions are less extreme. The invisible world outnumbers and outlasts the visible one. Microbes cover every surface, fill every drop of water, permeate soil. They generate half the oxygen we breathe. They recycle every element. They live in our guts, on our skin, between our teeth. We are ecosystems hosting microbial communities. Remove them and we die. Our relationship with microbes is not optional. It is fundamental. Understanding them reveals how life persists under extremes, adapts to change, and engineers solutions to challenges. The final surprise. In 2024, researchers at Montana State University used nanoscale secondary ion mass spectrometry, abbreviated NanoSIMS, to analyze magnetotactic bacteria consortia at single-cell resolution. They fed the bacteria isotopically labeled substrates and tracked which cells incorporated which elements. The results showed cells within consortia specialize. Some focus on carbon fixation. Others handle nitrogen metabolism. Still others manage phosphorus uptake. The division of labor mirrors multicellular organisms with differentiated tissues. But these are bacteria, prokaryotes, without nuclei. The discovery suggests multicellularity evolved independently multiple times, not just in eukaryotes. The magnetotactic bacteria consortia represent a separate origin of complex organization, dated to possibly billions of years ago based on magnetofossil records. Published July 11, 2024, occur in PLOS Biology by Schabel and colleagues. The work used single consortia metagenomics to sequence the genomes of individual consortia, revealing genetic heterogeneity never before documented in bacteria. Each consortium behaves like a single organism, but consists of genetically distinct, metabolically differentiated cells that never live apart. They divide together, swim together, die together, a multicellular prokaryote with no unicellular stage. That is the world microbes built. Ancient, diverse. Invisible to the naked eye, but shaping every visible thing.